should.
Definitely increase the degrees, like right when you walk into the room.
Yeah. It just smells like human bodies. It's strange.
A lot of humans in there.
Yeah.
I don't know how to describe things.
Yeah.
Awesome. And yeah, thanks again for setting up to do the presentation.
No worries. I was like, you know what? I might as well just get that done.
Yeah, I was going to say, I'm sure it feels really nice to have it over with.
Yeah.
Can you see if you're presenting?
No, no,
Some of the courses you can
Oh, yeah, sorry. I'm taking and calculus, multivariable calculus, and evolutionary ecology, and like the it's not bad.
Okay, let's get started again. So Dylan's going to say a few words about something called data camp that he alerted me to. So 60 seconds.
So this is data camp. Dolph was kind enough to sign us all up for it. And it's easy to walk through for learning R. It has anything from basic cleaning of your data to complicated stuff like ggplot, matlib courses. You can find all this information online by reading, but it's a little bit easier as it's interactive. And it'll give you kind of feedback on your code and your syntax. So I prefer learning this way than data diving on Google. But yeah, if you have a Gmail account, you can sign up with, I think that's what Dolph.
I might have to invite you or something to be
Yeah.
able to take the courses, but academic institutions can use the site for free.
Yeah, so free of charge until I guess the course is done, but if it's just another way of learning if you're interested.
Thanks. So the name gossip was mentioned and the name student was mentioned in the discussion. I just wanted to point out that they are one and the same person.
Yes.
So Gossett was a statistician who worked for the Guinness Brewing Company, and he couldn't publish under his real name. It has something to do with the fact that secrets got out from this company by previous employees, and they didn't want that to happen again, so he had to publish under the pseudonym student. Okay, so I'm going to just talk generally about features of experiments and experimental design. So some of this will be review, but I hope that in going over some of these issues, it'll make you think a little bit about how best to design experiments, if that's what you're going to do, and even design non-experiments, if you can't do experiments. First, I wanted to point out that you might not know it, but today is International Talk Like a Pirate Day. And what do pirates say? Exactly. And so according to Wikipedia, O'Reilly, the publisher O'Reilly, publishes a lot of R books. would have a discount on September 19th every year because it's International Talk Like a Pirate Day, so check that out immediately. But it seems like they've grown up in the meantime. I wanted to say a few things about planning your sample size. So that was not the topic of the discussion, but it was one that came up. How do you decide? And why is it important to decide? So I'll start there. One reason why it's important to decide is that ethics boards and animal care committees are becoming harder and harder in their requirements and want you to say things like, why is your sample size 30? Why is it? And you have to be able to say why it can't be 20 and why it can't be 40. And so justify why kill more animals than you need in order to carry out an experiment, and so on, and you know, why kill more humans than you need to if you learn, you know, with a smaller sample size, which treatment is the better. Shouldn't everybody be doing it sooner? Science is also expensive. Someone mentioned rats, like some of these rats, designed rats, you know, they cost $2,000 an animal, so you have to be pretty careful with those resources and try to get it right. And so the topic of designing a sample size sort of falls under the title of power analysis, although power is not necessarily the only thing that you are interested in. There's also a lot of talk these days in the literature about the low reproducibility of studies. You know, where an experiment will be carried out, a result is observed, someone else will repeat the experiment and find no difference. And I think and a lot of that can be explained by the fact that most experiments are underpowered. You shouldn't expect to see the same result again because you were lucky to see it in the first place. Low power in general has a high probability of a false negative. If there's a true effect, a low power study is less likely to detect it. Also, you know, in thinking about biological significance, as was mentioned, in relation to statistical significance, to me the way to think about biological significance is to think about magnitudes of differences. And that the estimation of those magnitudes is something that you get out of experimental designs as well. And the smaller your sample size, the lower the power of your study, the worse your estimates are. So if you want uncertainty to be low, then if you want your confidence intervals to be relatively narrow, then you know, sample size is something you need to think about, how to achieve it. Also, in a study which has relatively low power, any study, if you're lucky, yours, that finds a statistically significant result, the estimates of the magnitudes are likely to be exaggerated. And the reason is as follows. So imagine that there's a true difference between two treatments, the means of two treatments. A low power study means that some studies with relatively small sample size will estimate a small difference. Some will estimate a large difference. Which ones are going to be also found to be statistically significant? The answer is those that, by chance, you know, found an even, by the sampling error helped them find an even larger difference than is real. If you estimate magnitudes only on those significant studies, then those magnitudes are going to be exaggerated. Finally, it's not well appreciated, but that in a low power study, a high fraction of the results that are significant are in the wrong direction. And think about it in terms of a study where the magnitude of the difference between the two treatments is actually relatively small. Half the significant results, or nearly half the significant results, are going to be this way as that way. And in that case, nearly half the published studies which find statistical significance will actually be in the wrong direction. That's very counterintuitive, but it's nevertheless true, and that's something that may be more intuitive by the time we talk about meta-analysis, where we'll actually look at distributions of outcomes of multiple studies, all addressing the same question. So these are potential problems with low power studies. We think that even if we don't get a significant result, it should surely be in the right direction. Do not count on it. So when you think about sample size, it's important first to think about what exactly are you trying to do. And there are three or two main things that we're trying to do. And one is we're trying to estimate how big something is, how large something is, the slope of something, the magnitude of the difference between groups, and so on. And I think of that as a kind of a study or an experiment in which one of the goals is to measure the difference. How big the difference is really important. And in that case, the goal of designing or choosing a sample size is to plan for precision. You want to be able to get an estimate and to put confidence bounds on it that are not too wide. Because otherwise, if your confidence interval looks like this, the correct answer to your experiment, or the correct conclusion of your experiment is, I have no idea. Okay, that's what precision tells you. That's what confidence intervals are so useful for, to tell you just exactly how much information you have managed to obtain from your design, and it's good to think about this ahead of time, and to plan your sample size accordingly. You're probably more familiar with planning for power, trying to design an experiment whose sample size is sufficient, and often the idea is to choose a sample size that would have an 80% probability of rejecting a null hypothesis if the difference between the means was at least some value d. And so when calculating power, you have to specify d. And the probability of rejecting, you have to choose the power, and 80% is a conventional power. And there are formulas for doing this, and I'll introduce you to some of them in the lab this week, although we won't have a lot of time to try them out. Nevertheless, there are a lot of resources in R for calculating power. As was also mentioned, animals die, plants die, and when planning your sample size, it's also useful to think about the numbers of individuals that might die and to compensate as part of your planning. Then there are all kinds of decisions besides just what your sample size should be, because often if you're going to do an experiment, you might want to measure more than one thing, and then the question is, is it better to have more plots or more plants within plots? Is it better to have more families in your study design, or is it better to have a few families with lots of individuals per family? Those are the kinds of questions that you should be thinking about before you do your experiment. And then the question is, how do you decide those things? How can you make informed judgments? And we're going to try this in the lab this week. One of the things I would like to present to you, this package named R4, is that it's an extraordinary tool for just quickly doing a simulation to get a handle on, if the effect is this big, what are the advantages of having few individuals per family and lots of families, or the other way around? You could do this in an hour. Once this course is over, you ought to be able to do this. And sometimes the answers will surprise you. We did this for an experiment that was done in my lab on cold tolerance, and it turns out more families was better than more individuals per family when there was differences among families. And so we'll kind of just do a little bit of that in the lab this week. But when you plan sample size, you immediately have to specify things like, well, what size of difference am I trying to detect? Well, that's where biological significance comes in. D has to be a biologically significant number. And that's going to be very situation specific, very lab specific, very problem specific. But also, in order to calculate precision or power, you need to know something about the variability between your experimental units. How do you know that? Well, you can make an educated guess. Often that's the best you can do. And also, maybe someone has done a study similar to yours that gives you an idea of what that variability between units is, that give you an idea of what to expect. And also, particularly if it's an expensive experiment and little information is a variable, do a pilot study. Do an unreplicated experiment or a low replication experiment just to get an idea of what the kind of variability is that you're dealing with. Use that information to design an experiment with a proper sample size. Finally, we're going to read a paper for a discussion next week that talks about calculating power once your experiment is over. I will try to convince you that that is a totally useless exercise. And next week you should know why. So that's sort of planning sample size. Those are general issues, because beyond that, really, planning is going to be very specific to your particular problem and study system and so on. Now what I want to do is just talk a little bit about experiments and the features of experiments that you might consider when you design one. And some of this will go over issues that Hurlburt has raised. But what I'd like to do is maybe show you an example of what are essentially the gold standards for experimental studies, namely clinical trials. Show you one example and some features of it, and then make you think about, well, I wonder if really I should incorporate that feature in my experiment. First of all, what is an experimental study? Hurlburt talks about menscientific experiment, but there's no such thing. An experimental study, formally, the researcher determines which individuals go into treatment and which into control, and there has to be at least 2 treatments, or treatment and a control. So any experiment, like a large QTL analysis that all you're trying to do is map the locations of genes, they're often referred to as QTL experiments. They're not experiments. They're not experimental studies by this definition. You need at least 2 treatments, and the experimenter must decide who gets treatment, who gets control. In an observational study, nature assigns the units to treatment and controls. The researcher has no influence over which subjects receive which treatment, and that is really the essential difference between the two. And there are many other features, nevertheless, shared between experimental and observational studies, and so if you're carrying out an observational study, at least you can make use of some of those, even though you can't do the assignment of subjects to treatments, or treatments to assignments, treatments to subjects. Here's an example of an observational study and some of the features that make it different from an experimental study. And the main reason that, the main thing that this example will show is that the sort of the Achilles heel of the observational study is that it cannot distinguish between 2 possible explanations for an association between what I'll call an explanatory variable, which is the treatment variable, and then the response, what you measure. I'll turn off the light for a minute so you can see that these people are standing on top of Mount Everest. So Ray Huey at the University of Washington collected all the data that were available on climbs of Mount Everest and showed that people who climb Mount Everest are more likely to survive the journey if they bring along supplemental oxygen than if they do not. And a reasonable expectation is that supplemental oxygen actually increases the survival directly. But the other possible explanation is that supplemental oxygen has little effect. Survival and oxygen are associated because other variables affect both, like overall preparedness of the teams going up Mount Everest. Or maybe, the fact that more recent climbs have more access to technology that make this cheap, and so it's used more often. So there's all kinds of things that might be affecting both survival and whether or not supplemental oxygen is used, and that might be the reason we're measuring this association. Those other variables that distort the causal relationship between the explanatory and response variable of interest, oxygen use and survival, they're called confounding variables. And that's something that is the bane of observational studies, and the reason we do experiments is to get rid of them. So with an experiment, random assignment of treatments to subjects allows the researcher to tease apart the effects of the explanatory variable from those of the confounding variable. So if you randomly assign, you decide which climbing teams get supplemental oxygen and which climbing teams do not get supplemental oxygen, then your treatments will no longer be associated with overall preparedness because you've assigned them randomly. Overall preparedness will still contribute to variation among teams in survival, but that will be approximately equalized between the 2 treatments. And that is the principal advantage of experimental studies. Other than chance, oxygen, then, if there is an effect, oxygen is the reason if you do an experiment. Whereas that's hard to conclude if you cannot do an experiment, as in this case here. So we don't know really whether oxygen improves the survival. It probably does. There are good reasons for thinking so, but maybe the strength of the association is not as great as it seems. My understanding about climbing Mount Everest, which I've never done, nor will I, is that most people die on the descent rather than on the ascent. So they wait for a clear day, go up to the top, and then weather changes, and most people kill on the way down. So a clinical trial is an example of an experimental study, and more and more clinical trials are being used to judge the efficacy of particular treatment in the field of medicine. And the thinking about proper design of clinical trials, probably more thinking has been applied to clinical trials because the cost is so high in terms of human subjects. A clinical trial is just any experiment in which the subjects are humans. So experiments on non-human subjects are just called lab experiments or field experiments, depending on where they take place, or just simply experiments. So I'm just going to show you a quick experiment that was done. It was carried out on humans, and it was kind of an interesting one when I read about it for reasons that I'll show. So this was a study in which an experiment was carried out on female sex workers in Asia and Africa in order to look at a possible method for reducing the transmission of HIV. And they did a randomized controlled trial on this. And so there's a compound, a medicine, non-oxynol 9. Laboratory experiments in vitro had shown some activity against HIV-1. So enough background work had been done to motivate the far more dangerous and expensive act of carrying out an experiment on humans in the field. So what they did was they went to a bunch of clinics and they supplied this medicine to female sex workers, and then they measured as to whether their probability of acquiring AIDS or contracting HIV was changed. So the data were gathered on a volunteer sample of 765 HIV-free sex workers in 6 clinics in Asia and Africa. So there were 2 gel treatments, and they were assigned randomly to women at each clinic. So the female sex workers did not know which treatment they had actually received. Not only that, but the people handing them the gels also didn't know which workers were receiving the non-oxyl 9 gel and which were receiving a placebo that looked and smelled just the same. Okay, so nobody knew anything. Those that were actually involved. Here are the results of the clinical trial. So here are the 6 clinics in which the experiment was carried out, the sample size of each. And the feature of the experiment that I want to draw attention to is that the experiment was repeated in multiple places, and the approximately equal numbers of individuals were assigned to both treatments in each place. So basically the experiment was carried out, think of it as that the experiment was carried out 6 times in 6 different locations. And that provided them with substantially more coverage than would have been possible with just a single clinic. Anyway, the totals basically tell the story, which is that the treatment didn't work. And so it's not used. Which is amazing when you think about all the effort that went into this, and this goes on all the time. Huge amount of work, hugely expensive to do the background work, develop the medicine, do the background work, and then mount a clinical trial. Which, in this case, gave a fairly definitive answer. So let's just break down some of the components of this experiment that I think made it worth describing to you. And there are really sort of 2 design components that are involved. And one is the reduction of bias, the possibility that you will systematically deviate from what you're trying to test or estimate. And then the second is to reduce the effects of sampling error. Sampling error is all around us, but can we control it to a certain extent? How do we do this? So to reduce bias, the need for a simultaneous control group, I think, is pretty obvious to this crowd. So I needn't go into it, but a placebo is pretty much necessary. Randomization. So treatments weren't haphazardly assigned to women. They weren't strictly alternating in their assignments to women. They were actually randomized. And randomization is a great way to reduce bias in the design of the experiment. And then the third feature of the experiment to reduce bias is what's called blinding. And that is, the subjects didn't know which treatment they got. Neither did the clinicians who were handing out and interacting with the sex workers at the clinics knew which individuals were assigned which treatment. The people who actually talked to these women and assessed them and took blood samples and stuff like that, they had no idea which women were part of the treatment and which women were part of the control. So when the subjects don't know, that's called single blind experiment. And when the administrators of the treatments also don't know, that's referred to as double blind. And that's an important feature of clinical trials. Finally, sampling error. We know about replication. More is better, all else being equal. Balance is less important, but a relatively equal sample size is also increases power. And then finally, this feature called blocking, where subjects were grouped according to the clinic they attended. And so essentially you had multiple repetitions of the same experiment in different settings. So this paper came out in PLOS Biology a number of years ago, arguing that experiments on animals, and I think we should include plants in this too, that should be more like clinical trials, and that such experiments should involve blinding, they should involve randomization, and they should incorporate as many features as possible, because you might not be working on humans, but animals are also expensive, and so are plants, and that the reliability and repeatability of the study would be enhanced if everybody adopted as many of these features as possible. So that was sort of the argument of this paper. And part of their argument was to show, first of all, that a lot of the published articles out there on animal tissue samples or on rats, mice, and non-human primates, many of them didn't even state the hypothesis or objective of the study in the paper. You think this doesn't happen anymore. You're in a good school now. If I'm on your committee, this will not happen. But at the same time, you think, oh, well, that's totally natural, at least to state clearly what the hypothesis is. But here is a relatively recent accounting of how often that crucial step is actually not explicit in the paper. Few of the papers surveyed reported using random assignments of treatments to individuals, to subjects. How many of them used blinding? Very few, or at least very few said so. You'd think if you used blinding, you would mention it. So blinding is not very common in animal experiments, and perhaps it should be. We all know about the value of simultaneous control groups, but the only thing I wanted to mention here is that certainly when there's humans involved, there's a placebo effect. We all know that there's a placebo effect. It's measured over and over again. Not only that, even if you tell people that they received the placebo, there's still a placebo effect. Our brains are weird, what can I say? But the key feature is that the control subject should be perturbed in the same way. And I think that that's more realistic for the kinds of experiments that we do. So we probably heard about sham operation, in which basically the animals that are in the control are also operated upon, except the last step of the treatment itself is foregone. But if you're just doing field experiments and you're trampling all over the experimental plots in order to set things up, you should trample just as much on your control plots. That would be the equivalent of a sham surgery. So randomization, I've already mentioned, but I want to emphasize again that it really should be random, and that haphazard assignment, people are terrible at thinking up random numbers. It's been shown over and over again. There are apparent magicians who stand up there and guess the number that you're thinking in the audience. You know why they can do that? Because people choose the same numbers over and over and over again. Yeah, so as I said, the purpose of randomization is to break the association between confounding variables and the variables of interest. It doesn't eliminate the variation contributed by those confounding variables, it just equalizes them between groups. A simple design in which treatments are assigned randomly to experimental subjects is called completely randomized design. So that's the jargon. Blinding. Blinding is not something that's used a lot in experiments in ecology and evolution, but perhaps it should be. Blinding conceals information and thus prevents individuals from having their biases in some way influence the outcome of their, not just the outcome of the, the score or the measurement of the response variable. Yeah, that's what it is. Blinding is the process of concealing information so you don't provide information to the subjects and your own biases don't influence those results. Are you feeling better today? Are you feeling better today? The subtle changes in intonation can communicate a great deal to subjects in a clinical trial, and so it's better that doctors have no idea who's the treatment and who's the control. Only the statisticians should have complete information. So double-blind experiments are probably not so crucial if you don't work on human subjects, although people talk about their dogs. But blinding, in which you, the measurer, are unaware of whether the individual you are measuring comes from the treatment plot or the control plot, is actually a very good idea. And so the way to accomplish that is to use a system of numbered codes so that you are basically completely unaware of where the individual comes from, and that will reduce bias. This is a curious line. Studies showing that acupuncture has a significant effect on back pain are limited to those without blinding. When I thought about that, I thought, how do you perform acupuncture on somebody without them knowing it? Well, that's a good one. And I think maybe it has to do with, I don't know, where the needles are placed or something like that, or you stab them and remove the needle or something. But anyway, it's worth checking out this paper. So there are all kinds of, admittedly, observational studies showing associations between components of experimental designs that were left out and then the magnitudes of effects. So, medical studies without double blinding exaggerate treatment effects on average by 16%. That's an amazing result. Now you might think, well, that's not necessarily the effect of blinding by itself, because poor experiments are usually poor in multiple ways, and so it's hard to know which feature of a poor experiment is actually the main contributor to the exaggeration of effects and so on. But anyway, people have shown that there are certainly associations between presence and absence of these features in experiments and the magnitudes of effects that are found. Okay, minimizing sampling error. You'd like to make your estimates as precise as possible. You'd like to reject the null hypothesis if it's false. How do you do this? Well, one way is to control as many things as you possibly can. Certainly that's possible in a lab experiment. But another way is to use extreme treatments as well. So there are various sort of design features that are not so that are not at the statistical stage, but just simply deciding the conditions under which the experiment will be conducted. Admittedly, you'll be limiting, therefore, the generality of the outcome, but at least if there's an effect, you might have a better chance of detecting it. But replication is the first thing we think about when we think about power and when we think about precision, and that is, in general, the larger sample sizes, the better. They'll have, with larger sample size, all else being equal, you'll have a smaller standard error and a higher probability of getting the correct answer. Okay, so we already know this, and the purpose of today's reading, to read Hurlbert's essay from long ago, is really to think deeply about what replication actually means. And so I hope that you are impressed, and if so, I will make the students next year read the same paper again. So it's important to realize, and you will realize this after reading Hurlburt, that it's not about the number of individual plants or animals used. And really a better way to think about the experimental unit is that unit to which treatments are applied, and that unit which is interspersed. So clearly here's a situation, an experiment in which a treatment is applied to this plant and the control to that plant. We would all agree that is an unreplicated experiment. But it's less obvious to many that this is also an unreplicated experiment. And that's because the unit to which the treatment is applied, the unit which is interspersed, is not the plant but the chamber. And there's only one chamber for each treatment. Okay, and this is just your completely randomized design. We would all agree that this is a replicated experiment. So keep in mind that an experimental unit might be a single animal or plant if individuals assigned different treatments are interspersed. But an experimental unit is more typically made up of a batch of individuals, the fish in a tank, the plants near one another in a field plot, and so on, the colonies in a petri dish, the individuals in a household, and so on. Okay, and so the key feature is that if you treat the individuals belonging to the same unit as though they are independent replicates, then you commit the sin of pseudoreplication. And the reason it's a problem is because individuals in the same dish, the same tank, etc., are more likely to share other aspects of their environment and therefore likely to be more similar than two individuals picked at random from the population as a whole. I wanted to mention balance just briefly, but it turns out that the standard error is smallest for a given n when n1 and n2 are equal. So balance is a useful thing to try to achieve. It's not as important as replication. But if you can have it, it's good. And it has other benefits. I think of the whole lake experiment as a kind of one way to see the whole lake experiment is that it is a classic case in which there is no replication. Because, well, in this, the example that was shown, you've sort of fertilized one half and not the other. But if you can think of a whole lake experiment as being an incredibly unbalanced experiment, namely, N1 is 1, you're just going to fertilize one lake, but you have 10 other lakes that you might have fertilized but didn't, then you at least have replication in the controls. And so it is possible to some extent to get replication, even in the case of an experiment like a whole lake experiment, as long as the one lake that's fertilized is randomly chosen. And what you're basically trying to ask is whether then that one lake is an outlier relative to all the others. And if it is, then I think you can still draw some inference. Finally, blocking. So blocking was mentioned in the Hurlburt paper, but here's a way if you are carrying out a treatment that requires that your subjects be raised in chambers, then you can carry out both experiments in both chambers. And then chamber becomes a block. So a blocking variable is usually treated as a random factor in your analyses. And then once that's incorporated, then the effects of chamber can be somehow removed from the design and the treatment effects can stand out. So it's usually a good idea to block in general, particularly for field experiments. A familiar example of blocking is just simply a paired design. So that's something that Hurlburt mentioned as well. You know, if you want to know something about two treatments and plots, let's say the effect of clear cutting on salamander density. So you have two choices in front of you. You can just have a series of plots, some of which are clear cut and some which are not. Or alternatively, you can have a bunch of paired plots. Half of the plot, randomly chosen half of the plot is clear cut and the other is left standing. So those kinds of decisions you face anytime you sit down and ask yourself, how am I going to do this experiment? Whether you should be using a design with blocking, a paired design, or whether you should be using a two-sample design, or in the case of multiple treatments, more than that. And if there's variation, if there's heterogeneity from location to location, then the paired design can be a more powerful design. But that's what blocking means. But the key thing is that because this plot and this plot are not independent, they're side by side, that this must be taken into account when you analyze your data. And when you do so, you will avoid pseudo-replication. And randomized complete block designs is a paired design. You may have heard that term. It's a paired design. It's like a paired design, but the experimental units are 3 plots, like 3 treatments all in a group, and so on. So when there are more than 2 treatments in their group by block, we call it a randomized complete block design. Finally, another decision you want to ask is, if you're going to do an experiment, should you just vary one thing? Or should you vary more than one thing? Should you have 2 treatment variables that are crossed, as they would be in a factorial design? If you do, then you have the opportunity to measure interactions between. I just wanted to toss up one thing, and that is something that was brought up also earlier, which occurs when you take repeated measures through time of the same subject, such as plots in this classic experiment. So this was a Davidson experiment, and they carried out, they had these enclosures in, I think, the Chihuahua Desert, and they allowed rodents into one treatment and not into the other, and they looked at the effects on the number of colonies of ants. And each of these lines represents a trajectory of the number of colonies for a single plot. So the temptation is great to measure repeatedly, but if you do, then you have to include the individual plot as a factor in your design. And so that's how you can take account of the repeated measure. And by the way, repeated measures in time are problematic for another reason, called the sphericity assumption, which I'm not going to talk about right now, but I'll address in a future lecture. But just while you're thinking about designing your experiment, time is generally doesn't behave like other factors in experimental design, for reasons that I'll try to explain later. How to choose the best design for your purpose? It's going to be situation-specific, lab-specific, but I found this great package, Agricola, and what it does is it's a tool in R to help you model designs that are typically used in agricultural research, like how do you assign treatments to blocks in a randomized complete block design, so you can actually have it do the randomization for you as a way of helping you set up your experiments, and it'll also set up, it'll also do the randomization for more complicated designs like Latin square and so on, split plot designs. You can specify the randomization algorithm, you can specify the random seed in the pseudo-random number generator so that you can repeat it again. The package says that they generate information about parameters and so on, and will even draw a sketch showing the distribution of plots in the field. So if any of you tries that and you do this, I really want to see it, so let me know. That's the only tool I could find in R that actually maybe helps you think about what particular design you should use. So here's a point that was mentioned in other ways in the earlier discussion, and that is analysis should follow design. And this is one reason why it's a good idea to think about how you're going to analyze the data at the time that you're designing the experiment. So pseudo-replication, for example, is generally a problem of analysis and not design. It's possible to have a beautiful experimental design in which the treatments are replicated correctly, and then you can commit sacrificial pseudo-replication and lump everything together across plots, and thus carry out pseudo-replication. The way to avoid pseudo-replication is to remember that the analysis that you use should mimic the structure of the experiment. If your individuals are grouped by plots, then plot becomes something that structures your study design. Individuals within plots are groups, and therefore the grouping variable must be modeled as part of your analysis. So that's what I mean by analysis follows design. So usually those groups, like plot.
So do you mean if we do an experiment with like 2 rats in one box, we don't have to, we have to use the measurement as one individual. The box is one individual.
So rats in a box. So there's 2 issues with rats in a box. One is that the 2 rats are paired because they are located in the same, essentially the same plot. And so the design then would be presumably a paired design. If you have 2 rats in each of a bunch of boxes and one treatment is applied to one rat and not the other in each box, then that's a paired design. And in a paired design, essentially the boxes are the independent unit. In this case, doubly so, because if the rats can interact in such a way that they essentially contaminate your measurements by that interaction, then they're not independent for another reason. Okay? Another reason to think of the box as the unit of replication. So if the rats can't interact in a box, then you can include box as a grouping variable in your analysis that also includes the individual rat. But if the rats interact, then they really are not units at all. The box is the replicate. Does that make sense? So later we'll talk about ways to accomplish this in the tools that are available in R. And what it means when you include a grouping level variable like the box or the plot. Those factors are usually not of interest. They're just nothing that you have control over, and they're modeled as a random effect. And so what you're doing then, what you're trying to do when you're analyzing your data is carrying out, fitting a model to data that has both fixed and random effects, and that's called a mixed model, and we'll be showing you how to do mixed models in R. So recognizing how you'll analyze the data when you design your study is a really good idea. Because I don't know how you can calculate the power or precision or the sample size you would need in order to determine power and precision of your design unless you know how you will analyze those data. And it's a great exercise. And there are formulas for calculating power for simple designs like, completely randomized design with just 2 treatments. There's packages in R that will do that. But particularly when it's a mixed design with a grouping variable and so on, you might not find a formula for your particular case. I never even look at these formulas at all. I just prefer to go to R and make up data. Just invent some data, the kind of data that you think might be realistic for the kind of experiment that you're aiming to conduct, and then analyze it. And it's surprising how much intuition that will give you about the role of the number of groups, the number of subjects within groups, and so on, in thinking about what your sample size should be and how the individuals should be distributed among groups. Remember the question, how many individuals per family? Should you have more individuals per family, or should you have more families with fewer individuals? To me, that's an R question. You can easily test those alternatives with a quick simulation. And that's what we're going to do. We're going to sort of introduce you to that approach in the lab this week. The last thing I wanted to touch on was what if you can't do experiments? So remember, the one key feature of experiments is that there's the possibility of randomizing the assignment of subjects, treatments to subjects. But they're not always feasible. But fear not. We have made lots of really important and good decisions in the past on the basis of observational studies. So Ronald Fisher was very skeptical of the data that smoking cigarettes caused cancer. He was a lifelong smoker. And he was very skeptical because he thought, you can't do the experiment. To do a proper randomized controlled experiment on humans to determine if smoking causes cancer, you, the experimenter, has to decide who gets to smoke and who doesn't. That's what randomly assigning treatments to subjects actually means. If you want to do a randomized, if you want to do a proper clinical trial on the effect of smoking, then you have to do an experiment. And yet, amazingly, no one has ever done this experiment, and still we know that smoking really does cause cancer. So it's not necessarily the case that just because you can't do an experiment, you can't make progress. And I mean, there's various ways of making progress. In the case of smoking, you can make rats smoke and see if the outcomes are similar. So there are ways of doing experiments on non-humans that might carry over. And certainly we do experiments on non-humans all the time for exactly that reason. Nevertheless, so you can't randomize, but you can still employ a lot of the other features of clinical trials like blinding and so on. And then there are other steps that can be used in observational studies to limit the effects of confounding variables by matching individuals by their age and weight and so on. So there's a whole field that helps you do those kinds of things. Okay, so that's my spiel on sort of general thoughts on why we do experiments, how to think about analyzing your data while you're designing the experiment, and also to think about how to get information on the sample sizes that you need in order to carry out the experiment. So I have a couple of things on this last slide that I want to tell you about. The first is that this is the paper we're going to read next week, and I need volunteers. So I need 2 presenters and 2 moderators. Right, so my first is to say, this is what we're reading next week, and my second is to say I need some moderators and presenters for next week. And the paper is on what do you do when you get to the end of an experiment and your result is not statistically significant? What do you do? How do you move forward from that point? And one method that is or was commonly used is to do a post hoc power calculation. This paper is going to tell you why that's stupid and what you should be doing instead. All right, see you next week. And step up if you want to present next week. Otherwise, I'll use R's random number generator to choose.